Welcome back to The Explainer. Today, we're bringing you a focused clinical refresher on veterinary vaccinology, specifically zeroing in on delivery mechanisms and the absolute vital role of adjuvants in livestock health. If you're a veterinary professional looking to brush up on the latest in immunology and vaccine tech, you are in the exact right place. Let's get right into the science that keeps our herds healthy.
Okay, let's dive into this. Here's our clinical syllabus for today. We'll define the ideal veterinary vaccine, compare conventional approaches, explore next-gen delivery, unpack the power of adjuvants, and finally look at how we apply all this to livestock health. Now, before we get into the weeds of cellular responses, consider this number, greater than 50. That's the number of animal diseases we can currently prevent through vaccines. This sheer scale of impact is exactly what paved the way for monumental milestones in veterinary medicine, like the total eradication of rinderpest, a massive victory for cattle production and global food security. It's a powerful reminder of why we do what we do.
Moving to Section 1, the ideal veterinary vaccine. Let's establish our baseline goals. The ultimate wish list for vaccine development is pretty steep, right? The ideal vaccine should be highly immunogenic, induce long-lasting immunity, be completely safe for the animal, and remain stable even in harsh field conditions. Ideally, it's available in combined formulations, requires only a single dose, and of course, it's got to be affordable and accessible to all. Now, as veterinary professionals, you know this is the dream checklist. Real-world clinical medicine often requires strategic trade-offs between these different parameters. So the crucial point is understanding how the nature of the organism actually dictates our vaccine strategy. The type of immune response we need relies entirely on where the pathogen replicates. If an organism replicates extracellularly, we need an antibody response. But for intracellular pathogens, it's all about cell-mediated immunity. If the antigen resides in the phagosome, think Salmonella or Mycobacteria, it gets processed and presented by MHC class II molecules straight to CD4-positive T cells. But if it replicates in the cytosol, like Listeria and most viruses, it's presented by MHC class I molecules to CD8-positive T cells.
All right, Section 2, Conventional Vaccine Approaches. This is where we look at the safety versus efficacy balancing act.
There is always this classic balancing act. On one hand, live attenuated vaccines give us all the relevant antigens, providing a longer duration of protection and immediate localized immunity. However, they carry the risk of causing actual disease in immunocompromised animals, and you absolutely must maintain strict cold chain requirements. On the flip side, killed or inactivated vaccines offer massive safety benefits. There's zero risk of agent transmission and way less interference with maternally derived antibodies. The catch? They often give a delayed immune response, usually requiring a live primer. They offer no local protection, and individual application can get pretty costly.
Let's move to Section 3 and see how this builds into next-gen vaccine delivery, where modern genetic engineering lets us bypass those old conventional limits.
So modern approaches allow us to bypass older limitations using recombinant live vectored vaccines. The sequence here is incredibly elegant. First, we identify the specific immunogenic protein in the pathogen. Second, we pinpoint the precise gene coding for this protein. Third, we insert that gene right into the genome of a non-virulent vector, like E. coli or vaccinia virus. And finally, we vaccinate the subject using this recombinant vector. This brilliant technique was actually utilized by Professor Tilahan Nyoma in developing the modern rinderpest vaccine. The recombinant vector induces only a very mild infection while safely generating a robust immune response to the encoded antigen.
And this brilliantly illustrates the exact physical mechanism behind that engineering. We use restriction enzymes to essentially cut the DNA molecules, generating complementary sequences on both the vector DNA and the chromosomal DNA fragment that contains our target gene. Then, the enzyme DNA ligase swoops in and joins them together into a recombinant DNA molecule, which is then introduced into a bacterium. It's precise, it's targeted, and it completely eliminates the risk of disease transmission associated with wild-type strains.
Taking this a step further, we have nucleic acid, or DNA vaccines. Here, a plasmid DNA encoding a protective protein is delivered directly into the host animal cells. The host's own cellular machinery then directs the transcription and translation, effectively turning the animal into its own vaccine factory. Pretty amazing, right? But delivering this plasmid requires specific routes. We typically rely on intramuscular injection, transdermal delivery via a gene gun, or mucosal application using nose drops.
Now, the transdermal delivery is fascinating. A gene gun ballistically accelerates microscopic gold beads, each coated with about 600 copies of DNA plasmids, literally shooting them straight through the stratum corneum and into the epidermis. Once they're in,
Resident dendritic cells, specifically Langerhans cells, gobble up these beads. As those antigen-presenting cells migrate through the afferent lymphatics over to the draining lymph node, the foreign DNA is expressed, degraded into polypeptides, and presented right alongside MHC molecules. This kicks off T cells and subsequently B cells. It's an incredibly targeted way to jumpstart both the humoral and cellular responses.
Moving right along to Section 4, the power of adjuvants. Time to talk about those essential immune modulators. By definition, an adjuvant is a substance incorporated into or injected simultaneously with a vaccine for the express purpose of potentiating or modulating the immune response. Because conventional dead vaccines, toxoids, and purified subunit vaccines lack the natural replication and native danger signals of live pathogens, they almost always require these immune modulators to actually function properly in the host.
The heavy lifting that adjuvants do really cannot be overstated. They increase the overall intensity of the immune response, which in turn lets us significantly reduce the physical quantity of antigen required per dose. Adjuvants also prolong the duration of immunity by creating a depot effect, meaning they slowly release the antigen over time so it isn't rapidly eliminated. Plus, they can stimulate specific response types, shifting the bias towards cellular or humoral immunity depending on what the specific pathogen demands. Ultimately, they reduce the necessary frequency of booster immunizations. They are, for sure, the critical bridge between a short-lived antibody reaction and practical, lasting herd immunity.
And that brings us to Section 5, Adjuvants in Livestock Health. Now, what's really interesting about this next bit is how we apply this theoretical pharmacology directly to field livestock.
Let's anchor this theory in a clinical reality. Take the vaccine for contagious caprine pleuropneumonia, or CCP. Without an adjuvant, a 1.5 milligram per milliliter concentration of the vaccine just could not adequately protect against clinical disease, and immunity lasted only about 6 months. But by simply adding a sapamine adjuvant, the required antigen concentration dropped drastically down to just 0.15 milligrams per milliliter, while the duration of protection doubled to greater than 12 months. This makes the vaccine significantly cheaper to produce and vastly more effective out in the field, a real game changer.
For rapid professional reference, let's quickly summarize the big players you'll encounter. Aluminum-based adjuvants create that depot effect we talked about, pushing a T2 humoral response, but they can occasionally cause contact hypersensitivity. Friends Complete Adjuvant uses an oil emulsion with heat-killed mycobacteria for massive cellular and humoral stimulation, but you gotta watch out for the notable risk of chronic local inflammation. Saponin, derived from natural plant products, triggers strong cellular responses, but can cause local reactions or hemolysis. And finally, IceComs, or immunostimulating complexes. These use a really unique cage-like structure of cholesterol and phospholipids to hold the antigen, inducing long-lasting cell-mediated and mucosal responses with very minimal side effects. This brings us to a crucial question for the future of our field. With continuous advancements in recombinant vectors and precision adjuvants, which stubborn livestock disease will veterinary medicine eradicate next? As we've seen today, from targeting precise cellular pathways to fine-tuning the depot effect of our vaccines, the tools at our disposal are becoming remarkably sophisticated. Thank you for joining me for this explainer. Keep this knowledge handy in the clinic and out in the field, and I'll see you next time as we continue to break down the science that drives veterinary medicine forward.